So welcome back. We're going to dive right into where we left off. And so for the rest of this lecture, we're going to talk about some of the structural proteins that are involved with muscle contraction. And then we're going to get into kind of the nitty-gritties on how the message is basically transferred from the nerve and a nerve signal, an electrical signal down a neuron, to the actual muscle itself. So really cool lecture in terms of it's amazing how much occurs at that neuromuscular junction and how many steps are involved, and how, again, this is where I think anatomy is amazing, that there's so few instances where it goes wrong. So it's just pretty amazing. So hopefully you find it as fascinating as I do, and let's dive right into structural proteins. So we talked a little bit about some of these structural proteins before, but I kind of just grazed over them. We barely mentioned them, and now we're going to really get into what they actually do. So we have these structural proteins inside these myofibrils that are going to really be there to help keep the thick and the thin filaments in their proper alignment, because it's really important, obviously, for the contractile activity of these sarcomeres for everything to be in a line. So the structural proteins will also help to give the myofibrils elasticity and extensibility, right? So we want, again, them to be able to extend, and then obviously to be able to return to the original state. Again, elasticity is their ability to return to that original state. Extensibility is their ability, again, to be stretched. And then lastly, to link the myofibrils to the actual cell, muscle cell membrane called the sarcolemma, as well as the extracellular matrix outside the cell. So again, we're going to talk about all of these structural proteins here in a little bit more detail. I'll actually put this in presentation mode for you there. So the proteins of, again, muscle, some of the proteins of our sarcomeres, really. And the first big one that we're going to talk about is titin. And just like it sounds, it is the titan of muscles, just spelled T-I-T-I-N instead of T-I-T-A-N. And it is this filament that's going to anchor basically these thick filaments here to the Z line. So it's going to run here. You'll see titin's going to run kind of the whole length here of the sarcomere. And it's going to run basically, or it's going to anchor, I should say, itself in the M line and then at the Z lines here, the Z discs here. And so what this protein is going to do is it's going to act kind of like these guys here, like the shocks, right, in your car, and or spring in anything that has a spring in it, right? And basically, what it's going to do is that protein can actually stretch to about 4 times its resting length, and then spring back, right? Because that's what it would do as a spring back to that original state unharmed. So again, this is what helps to allow that sarcomere to do that. And again, I like to picture it, just like, the springs and the shocks of your cars or your SUV, okay? And this is really important because obviously this plays a big role in the recovery of the muscle after it's being stretched. Again, obviously to a point, if you overstretch a muscle again, then you can damage some of this and even, possibly tear this from the actual Z disc. But again, for the most part, in normal muscle contraction, when a muscle is stretched, this protein is what helps it kind of bounce back, right? Kind of spring back to its original state when that muscle relaxes. So again, titin is a really important protein, kind of the titan of proteins. Haha, my horrible pun there for you, but it really is kind of the titan of proteins there in terms of the proteins in the sarcomere. And then you have on the M line here, you have this protein called myomycin. And so myomycin, nice, it starts with the M, it's on the M line, right? Kind of helps you out there. But it's going to help kind of connect, tighten and hold tighten in place, and as well as these thick filaments in place. And so it's going to kind of link these thick filaments and tighten and kind of help keep and maintain their alignment. So again, important, and that's what forms that M line, is this myomycin. Nebulin, which is a protein that you can't see here, is an inelastic protein, meaning it's not elastic, and it's just going to wrap around the actin filaments here and help to kind of keep them in alignment. But again, you can't see that here. And then dystrophin, which you may have heard of before. So if you've heard of muscular dystrophy, you may have heard of this protein. But dystrophin is going to, and again, not depicted here, but it's going to link the thin filaments here to the sarcolemma. So that's why it's not depicted here, because you can't see the sarcolemma. But it would link these thin filaments to the actual cell membrane, the sarcolemma. And then it helps to transmit the tension, right, from these sarcomeres to the actual tendon. So from these myofibrils eventually to the tendon. So really important. And I'm sorry. So when I say, again, transmitting that tension, remember that tension would be transmitted from the myofibrils and then eventually again to the sarcolemma of that muscle fiber and then out to the tendon. So really important protein, and so important that if for some reason you have a genetic defect in this protein, and it's actually an X-linked trait. So unfortunately, again, you see this primarily in men, because remember that anything that's carried on the X chromosome, if you're female, you have two X chromosomes. So you have another shot, right, at getting another normal form of this gene. So if you can get a normal form of this gene, then you can make the normal dystrophin. But if for some reason mom passes on this, muscular, she's a carrier and she passes on this muscular dystrophy gene, then if you're a male, you don't have another X chromosome because you just have Y. You don't have that other X chromosome again to give you another shot. So you end up with the genetic defect that codes for a form of dystrophin that isn't functioning. So basically it can't function properly. So again, this genetic defect in the gene that codes for dystrophin that doesn't function properly causes all kinds of issues, because then without the dystrophin, right, there's no protein to transmit that tension to the tendons. And so what happens is all of that tension gets put on the sarcolemma. And what it happens is then it ends up causing tears in the sarcolemma. And as you can imagine, this is tears in the cell membrane. So anytime you have tears or holes that are forming in the cell membrane, that's really bad, because it can let things like calcium, right, out of the cell. And now you no longer have calcium, available to be able, like we just talked about, to bind to the troponin, right, and allow for muscle contraction. So again, this causes all kinds of issues, and eventually the muscle fibers just die. They end up dying off with all of this wear and tear on the sarcolemma. And then eventually this leads to the atrophy of muscles. So basically the muscles will just start to atrophy and die off. And so you can see this is obviously a really severe case where you can see, again, he's had severe muscular atrophy here. And then again, you oftentimes end up with, kids, this is where kids end up in wheelchairs and things like that, because they, and again, this is the more severe Duchenne's muscular dystrophy. There's different forms of muscular dystrophy, but this is Duchenne's muscular dystrophy, or DMD. And so again, it's progressive with age, and unfortunate because there really aren't a lot of great treatments for it as of yet. So, and this is kind of because again they show the genetic link here for Duchenne's muscular dystrophy. So. Okay, so we're going to go ahead and talk a little bit about this length tension relationship. And this is just the fact that when we talk about muscle contraction and the force of muscle contraction, it depends actually on the length of the sarcomeres before muscle contraction begins. And so they have done research on this, and they found that there's kind of like this sweet spot, right? So this ideal spot, this optimal sarcomere length prior to muscle contraction that allows for the most force to be generated, the most tension, right, to be generated. So this muscle, again, can contract fully. So to get kind of the most bang for your buck, you have this kind of optimal sarcomere length that's operating at, again, about 80% to 120% of its resting length. So here you can see if the sarcomere is really contracted, really short, before you even start muscle contraction, there's nowhere else for this to go, right? Like this sarcomere is already as short as it can go, right? So you're not able to generate, any force from that. And then now on the other extreme end here, if the sarcomere is excessively stretched before you even start muscle contraction, then you don't have enough myosin heads here really able to like generate that tension for muscle contraction. So you need to have, some, that optimal, basically, overlap of the myosin heads and actin to start. But again, you don't want to have the sarcomere too short either. So this is that kind of optimal sweet spot there. So how does a motor neuron actually, and this is where it gets really interesting. How does a motor neuron actually get a muscle fiber to contract? Okay, and so what's the process behind how this happens? And so all of this is going to take place at this spot called the neuromuscular junction, or you'll hear it abbreviated NMJ. So again, N for neuro, M muscular, and J for junction. So this basically what is going to happen here is as a message is transmitted down a motor neuron, okay, there's going to be an electrical signal that's going to move down and wash over the membrane of the neuron. And when it reaches the very end of this, that we're going to call this synaptic bulb, but what we call the axon terminal. So it's going to come down that axon terminal and then reach this, what we call the synaptic bulb. And at the synaptic bulb is where it's going to kind of pass this message in the form of a chemical messenger across this tiny gap to this spot on the actual individual muscle fiber, okay, or muscle cells. Remember, muscle fibers, muscle cell. So this is all going to take place at what we call this neuromuscular junction, or you'll sometimes hear it called in some other textbooks, they just flip-flop it, the myoneural junction. Again, muscle, neural meaning, again, that neuron junction. So again, we're going to use here in this class and with this textbook, neuromuscular junction, but I like to show you just in case you hear it in another forum in another class. So again, neuromuscular junction, myoneural junction mean the same thing, that junction between a neuron and a muscle cell. And so a couple of bits of terminology here. So the end of the axon here is what we're going to call the axon terminal. And that axon terminal is going to literally terminate at what's called these synaptic end bulbs. So this kind of, this area that if you look here, kind of forms this bulbous area at the very end of that axon terminal. And so that's the synaptic end bulb. And we'll talk about kind of why it forms that shape in just a minute when we talk about its function. So here's your synaptic end bulb. Now, believe it or not, this synaptic end bulb actually isn't in contact directly with the sarcolemma of the muscle fiber. So there's actually a little bit of a gap. And so we call that gap the synaptic cleft or the synaptic gap. Okay, so there's a slight gap, and you can kind of see that slight gap here. And then what ends up happening is it's going to, again, like I kind of talked about, it's going to pass this message on across this synaptic cleft by releasing something called a neurotransmitter that's going to help transmit the signal. It's basically a chemical, okay? And we'll talk about even in more depth the fact that that chemical is called acetylcholine, okay? That's our kind of neurotransmitter. So what's going to happen is it's going to transmit this message using this chemical messenger, and on the sarcolemma here, there is going to be a spot here that's going to kind of mirror the synaptic end bulb, and it's going to have a bunch of acetylcholine receptors, so a bunch of these neurotransmitter receptors on it. And so because this part of the sarcolemma membrane is very specialized with all of those receptors for the neurotransmitter, we actually give it a special name. We call it the motor end plate. So this motor end plate is actually this region that's going to accept that neurotransmitter on the sarcolemma. So it's just a part of the sarcolemma membrane of that muscle fiber. So again, just a little bit of terminology, so that way then as we go through this, hopefully that will help you out a little bit. So I'm going to go through that again here. So when you look at this, this is again kind of zooming in here. So here you can see, here's your motor neuron coming in, and then here's your axon terminal ending in this synaptic end bulb. Now here, with the synaptic end bulb, you're going to see there is this synaptic cleft or synaptic gap here. So there's a little bit of space here between that motor neuron and the sarcolemma membrane of the muscle cell. Now, if you look here, the sarcolemma membrane is going to be kind of folded in here. And you're going to see there are lots of little receptors here on this membrane. And so this is what we are calling the motor end plate. This right here, all of this is the motor end plate. And the reason, again, this is so important is that it's the reason that we are able to transmit the messages from that neuron across that synaptic cleft. Okay, so again, this here is the motor end plate. So now, again, we talked about synaptic end bulbs, again, are these swellings of the axon terminals, and the end bulbs are going to contain, and the reason that the synaptic end bulbs are kind of bulbous shaped like this is they're going to have a bunch of these vesicles inside of them that we call presynaptic vesicles. Because they're, before synapse is, again, the joining of these two. So synapsing is joining of those two without kind of passing the ability to pass on a message without actually touching, okay? And so, again, these are going to be filled with vesicles of neurotransmitter, lots and lots of vesicles of neurotransmitters. So that's why it's bulbous shaped. And these synaptic vesicles are going to be filled with a very special neurotransmitter called acetylcholine, okay, which we're going to abbreviate as ACH. So hopefully you'll kind of get familiar with that. It's just acetylcholine's a long word to write out, so we abbreviate it ACH, okay. And then if you look at this motor end plate, the motor end plate membrane actually contains up to like 30 million acetylcholine receptors. So lots and lots and lots of acetylcholine receptors. So again, lots of acetylcholine being released, lots of acetylcholine receptors, again, to receive that message. So let's kind of talk about what's going to actually happen here. And this is a pretty good kind of zoomed in diagram. Again, you can see all of the same things. Here's this, the synaptic bulb here. At the end of the axon terminal, you can see all of these presynaptic vesicles with neurotransmitters, specifically acetylcholine inside of them. Sorry about that. And then you're going to see here on the motor end plate of the, again, the motor, or the muscle fiber. So again, this motor end plate is just this kind of folded region of the sarcolemma of the muscle fiber itself, okay? So again, if you need to stop and kind of process this for a moment, you can pause this video and kind of process these terms, because now we're going to start to talk about how this process happens. So what's going to happen is here, you're going to have a message that's, again, going to be, it's an electrical signal that's being passed down this neuron, right? And when it's received, that basically that electrical signal washes over the membrane down at the axon terminal and then at the synaptic bulb, okay? It's going to actually cause the release, it's going to stimulate the release of this acetylcholine from that synaptic, from these synaptic vesicles at this synaptic cleft here. So then that acetylcholine, okay, is going to be released into the synaptic cleft, and it's going to move across that synaptic cleft, and it's going to bind to these acetylcholine receptors on the motor end plate. So here you can see it's binding here to these channels, okay? So now these receptors that it's binding to are actually gated ion channels. And when acetylcholine binds, sorry, I don't know if you can see my, there, you can see my arrow now. But when that acetylcholine binds to this gated ion channel, it's going to trigger it to open. And that's going to trigger an influx of sodium. And that sodium is going to rush in it. And you have to, again, remember that there are, again, millions, billions of these acetylcholine receptors here. So again, you have millions of these channels, these gated ion channels opening up to allow sodium to rush into this cell, okay? And as that sodium rushes in, it's going to make this muscle cell more positive. And this is going to trigger a muscle action potential now. And now, so as that sodium rushes in, it's going to cause, and eventually we'll talk about this a little bit later on, but it ends up causing a change, an electrical change in the charge inside this cell. And as it does that, it's going to trigger these other electrical gated ion channels to open up, causing more sodium to basically rush in. And so this is how that, you don't have to know all those details yet, but I'm just kind of giving you that. We're going to get to that more when we get into nerve conduction. But basically what you have to know here is that is going to trigger this action potential to be propagated across the whole entire muscle fiber, okay? So then, as this is propagated across the whole entire muscle fiber, sorry, I'm going to go to this image because this just is a little bit of an easier image to show this. So here you can see this is where that action potential, again, was generated and now is washing over and propagating over the entire muscle fiber membrane, across the entire sarcolemma membrane. And as it is, right, this electrical message, this electrical signal is going to be transferred across the sarcolemma and down into these T tubules, okay? Now, as it moves and travels down into these T tubules, it's going to signal the release of calcium from the sarcoplasmic reticulum, okay? And so as it triggers that release of that calcium from the sarcoplasmic reticulum, the calcium is now in the sarcoplasm. And now that calcium can go and it can bind to that troponin, right? On the troponin-tropomyosin complex, pull that tropomyosin off of that myosin active binding site, right? And then that allows that myosin head then to make contact with that actin, that active site on the actin, and then that's going to cause that ratcheting. And that's where you're going to get, again, the shortening of that sarcomere in terms of muscle contraction. Okay. Now, then what ends up happening afterwards is there is an enzyme in here called acetylcholine esterase. And I know I have this in a further slide spelled out for you, but acetylcholine esterase, which we typically write, because we abbreviate things, right? It ends up being these really long words. So oh my gosh, I'm horrible with this pen. I really have to apologize. Acetylcholine esterase is going to be ACH, and then it's abbreviated ACH and then E for the esterase. But esterase, again, it's an enzyme. And so this is the enzyme that actually is going to degrade acetylcholine, because you don't want the acetylcholine constantly hanging out in that synaptic cleft, causing muscle contraction when you want muscle contraction to slow down, okay? Or you're done with the muscle contraction for that muscle. So acetylcholine esterase is going to break down this acetylcholine in that synaptic cleft, and then basically that will allow the muscle action potential to cease, and then the muscle cell can finally again return to its relaxed state. So that's kind of the whole big picture here, in terms of what's happening. And let me go back here a few slides. Oops. In terms of muscle contraction. So this is my kind of like step-by-step process for you. So that is kind of all written out for those of you that like that. But again, and then here is kind of the visual of this, okay? And so I've got, again, a couple of visuals. I liked this one too, so I put a couple of them in here because I feel like, again, it's just helpful to see the whole kind of big picture thing, but some of them are zoomed in a little bit better. I liked this one because this one really shows that the sodium and the potassium channel here, ion channel. Again, we're gonna focus mostly on the sodium coming in, but it also allows potassium out. And it's good to just kind of get, even though I know we don't really talk that much about potassium here, but it's a good idea to kind of get used to this idea, because this is what we're going to talk about when we talk about nerve conduction. So if you can think about, again, sodium's going to be rushing in anytime we're spreading an action potential. Sodium's going to rush in and potassium is actually going to come out. And we'll talk about why this is, again, when we get to nerve conduction in the next few chapters. But for now, again, it's just a good idea to know sodium rushes in, potassium rushes out as that action potential, again, hits and propagates down the membrane. Okay, so again, just to kind of review one more time, action potential coming down the neuron, boom, it hits this synaptic bulb and basically signals, right, the release of acetylcholine from these synaptic vesicles. So then acetylcholine travels across the synaptic cleft, binds to the acetylcholine receptors on the surface of this motor end plate, causing these sodium-gated channels, ion channels, to open up. Sodium rushes in and some potassium rushes out, and that is going to cause, again, this change in what we call the membrane, the electrical membrane potential of this area of the membrane. And that is going to stimulate the opening of more of these gated ion channels, propagating this action potential down, again, that sarcolemma. Now, as it propagates down that sarcolemma, it's going to propagate down these T tubules, stimulating the release of calcium that's stored. Remember, the sarcoplasmic reticulum is going to store, that's one of its main purposes here in muscle cells, is it's going to store calcium. So then it's going to release that calcium. That calcium is then going to be free in the sarcoplasm. It's going to move through the sarcoplasm and eventually find a troponin molecule to bind to. When it binds to that troponin is going to undergo a conformational change and pull that tropomyosin off of that active site, exposing that active site for the myosin head now to bind and form that cross bridge. And as soon as it does, it's going to undergo that power stroke, shortening again that sarcomere. Because remember, those myosin heads are creeping along that sarcomere. Okay, so again, I think it's helpful to have a few animations to put this. I'm a very visual learner. I think it's very helpful to have some of these animations. So please take the time, pull up my PowerPoint, click on these links, and watch these animations. This one does a really good job of focusing on just what's happening at the neuromuscular junction. This one then kind of takes that to the next step and shows you how that, again, is going to impact that shortening of the sarcomere. And then this one kind of takes you through the whole process and puts these two all together. So again, let me know if those animations are helpful, or if for some reason you are just still really struggling with this and it just doesn't make sense to you, then please see me. The other thing that I suggest that you do for this is, take either the step-by-step process if you like that, or, print some of these images off. So like this image here, maybe print this image off and stick it on your mirror. Again, this is one of those things where you're brushing your teeth twice a day. And so go through this whole process in your mind. And if you can do that, then you're going to be just fine for the exam in terms of this. Okay. So those are a couple of my study hints. Again, let me know if they help you and if they work out for you. If not, again, I'm here to help and we'll try some other things if that's not working for you. Okay, so now let's get into some really interesting stuff. Let's talk a little bit about the pharmacology of the neuromuscular junction. And this gets interesting because now we can talk about some of the stuff that you're really interested in terms of why you need to know what's going on at this neuromuscular junction. So some of you have probably heard of the botulinum toxin. Again, this is oftentimes you'll hear about this in terms of botulism if you've eaten some bad contaminated food that's been contaminated with this bacterium. And again, we don't have this happen all that often anymore, but this used to be more of an issue when we had like foods that were improperly canned. And so what ends up happening is this bacteria called Clostridium botulinum produces this nasty toxin. And it's called botulinum toxin. And that botulinum toxin actually blocks the release of the acetylcholine neurotransmitter at that neuromuscular junction. So if you can't release acetylcholine, right, into that synaptic cleft, you can't do muscle contraction. And so what ends up happening is this causes oftentimes paralysis of many muscles, but one of the most important muscles being, again, your diaphragm. And if your diaphragm, right, can't contract, then you're not able to properly do respiration and bring, again, air into your lungs. And so death oftentimes occurs due to paralysis of the diaphragm, and that person can't breathe anymore. So that's kind of an interesting one. Another one is Black Widow, not that Black Widow. Sorry, trying to keep you going here. But this Black Widow. And Black Widow spider venom actually works in a really interesting way. Initially, what ends up happening is Black Widow spider venom actually is going to increase the release of acetylcholine. So it's going to stimulate the release of lots and lots of acetylcholine across that synaptic cleft. So it's doing just the opposite of what the botulinum toxin's doing. But what ends up happening is this first initially leads to really painful muscle spasms and rigidity. So your muscles are all, again, contracted and spasms, not all of them, but oftentimes, again, a lot of times individuals that are actually bitten by a black widow will have some of their muscles, again, undergoing these spasms and rigidity. And then this is typically followed by kind of a progressive kind of failure of the transmission of those messages at the neuromuscular junction, because those presynaptic vesicles are going to be all depleted. Basically, you're going to run out of acetylcholine, and now you don't have enough to do kind of the normal muscle contraction. And so then it ends up actually causing paralysis as these presynaptic vesicles are depleted of their acetylcholine. So initially it causes muscle spasms and then eventually causes muscle paralysis when those presynaptic vesicles are all depleted of their acetylcholine. So really interesting, different way in which it works. And then this one you may have heard of. It's called curare. And you may have heard of this because it's actually used oftentimes to relax muscles during surgery. But it is actually a plant poison. It's from the rainforest, called, again, a plant called curare. And they initially discovered it because some of the initial tribes in the rainforest, the Aboriginal tribes would use this curare from this poison from this plant, and they would put it on the tips of their arrows so that they could use it again when they're hunting. And what happens is curare ends up causing muscle paralysis by blocking the acetylcholine receptors. So it actually blocks, instead of, again, like botulinum toxin, which blocks the release of acetylcholine, here it actually binds to and blocks these acetylcholine receptors. So if acetylcholine can't bind to those receptors, then again, it can't stimulate muscle contraction. So again, in just a different way. It's cool because these three all work in different ways. This one, again, is going to block the presynaptic release of neurotransmitters. So it blocks, again, the release of acetylcholine. This one here initially causes the too much release of acetylcholine, and then you run out of acetylcholine. And this one here blocks at the acetylcholine receptor. We call it the postsynaptic, on the other side of that synaptic cleft. It's going to block those acetylcholine receptors. So kind of cool because they all three work in different ways. And then here also, snake venom, it depends on the snake and the type of venom. Some types of venom will actually block your release of neurotransmitter, what we call the presynaptic, again, before the actual synaptic cleft. And then others can interfere with the postsynaptic acetylcholine receptors. And that's what this guy does. So coral snake venom actually inhibits the acetylcholine receptors and prevents, again, the binding of acetylcholine to those acetylcholine receptors, a little bit like curare. Okay, and then that causes, again, that kind of basically that neurotoxic muscle paralysis. So again, really interesting in the different ways in which they work. So this is just a slide. It's basically what I just talked about there in terms of all of the different ways that some of these toxins can work. And again, some of them we use to our advantage, like here, again, using curare as a muscle relaxant during surgery, so that, again, you can have those muscles won't be in a contracted state while you're, either trying to like cut through the muscles to get through to different anatomical features and whatnot, or move those muscles out of the way. So again, some more information on rigor mortis. I already talked about rigor mortis, so you guys kind of already know this. It's just a slide on this. But basically, again, it's to remind you that ATP, again, is needed for cross-bridge detachment for, again, that myosin head to be released. So again, if for some reason, again, when someone dies, then they're not producing ATP anymore. And this, again, is going to result in those myosin heads staying bound to the actin and causing this kind of constant state of contraction, which we call rigor mortis. This typically, again, happens about 3 to 4 hours after death. And then, again, those muscles really start to stiffen. And so peak rigidity, they say, occurs at about 12 hours post-mortem. Which is really interesting that it takes that long. And then the only way that, again, these muscles, these rigor muscles start to kind of relax is when you literally get just the decomposition of the proteins in the sarcomeres, in the myofibrils. And so eventually, as those proteins start to break down, then the myosin heads drop off as they start to break down and the actin starts to break down in terms of decomposition. So the other interesting thing is that intracellular calcium levels will actually increase because ATP is no longer being synthesized. So the calcium can't be pumped back into the sarcoplasmic reticulum. So we haven't really talked about, I don't think we really even talked about that, but after muscle contraction, again, after you stop getting the signals for muscle contraction, you're no longer getting the signal from that motor neuron for that muscle cell to contract. What ends up happening then is ATP is used to pump that calcium back into the sarcoplasmic reticulum. Well, if you're not producing ATP, then you can't pump that calcium back into the sarcoplasmic reticulum. And so again, that calcium is going to stay out there in the sarcoplasm and it's going to be free to bind to that troponin tropomyosin complex. So there's kind of two real reasons here in terms of why we get rigor mortis. And again, both are due to, of course, the lack of ATP. So just, again, kind of interesting. So since we're talking about ATP, let's real quick talk about muscle metabolism and the production of ATP in muscle fibers. So again, obviously, as you probably know, sorry, I'm going to get a drink of water here, just like I would in a real lecture. But as you probably know, again, muscles are going to use a lot of ATP at a really high rate, right? Especially when they're active. And the sarcoplasmic ATP, again, is going to last only for a few seconds. That means the ATP that's just normally, running around in the sarcoplasm. Remember, the sarcoplasm is the cytoplasm of the muscle cell. And so we have to have these other extra sources of ATP production, obviously. So one of them is one that you may or may not have heard of from 181, creatine phosphate. And then the other two you should know about: anaerobic cellular respiration and aerobic cellular respiration. Those are kind of the two ones that you've probably heard about. Now, please keep in mind that I am not going to have time to go over anaerobic cellular respiration and aerobic cellular respiration with you. So again, we're assuming that you had this already in Bio 181. And so what you need to know is at the level that we're going to talk about in these slides. So meaning I'm not going to go back through your 181 course and make you dig up some of the details of that, okay? So we're assuming that at least you know the basics on anaerobic. Again, means doing ATP production without oxygen, and then aerobic is obviously with oxygen, okay? That oxygen requirement as that final electron acceptor. I know you guys are like, no, but at least, again, that gives you kind of the basics again. And like I said, you just have to kind of know for our purposes, we're going to talk about how this is important in terms of ATP production and what that means for the muscle fibers. Okay. So at the level that we're going to talk about here. So creatine phosphate, again, is going to be kind of your first place that you're going to turn to as a muscle fiber. And creatine phosphate can basically power up your muscle contraction for, again, about 15 seconds here. So this is really for short bursts. Again, this is great for your 100-meter dash. And so basically what creatine phosphate does is it's going to recharge your ATP. I like to think of it as ADP is your rechargeable battery, and creatine is going to be the recharger. It's going to be the charger. And so what's going to happen here, and again, this is really good for just short supplements. This is kind of what I picture again in my head, right? Is that as you use that ATP, remember, when you break down ATP to ADP and phosphate, that's when the energy is released, right? For muscle contraction. So then creatine phosphate is like your charger, right? Recharging the battery. So now when you have ADP, it's your dead battery, right? And you need the energy, okay, from something to give you that extra phosphate to recharge that battery. So that's creatine phosphate. Creatine phosphate's there to give you that extra, or give ADP, I should say, that extra phosphate, recharging that ATP battery there. Okay, so now you need that creatine again to form creatine phosphate. So again, once creatine phosphate is used to add on that extra phosphate to your ATP, then creatine could then be recharged by another ATP, okay, eventually. So again, when you're not using up all that creatine, then you're going to recharge all of your creatine batteries, their batteries themselves, right, into creatine phosphate. So what ends up happening is athletes actually try to take in more creatine in the hopes that this will allow them to contract their muscles at a higher intensity for a more prolonged time, right? Because if you have more creatine, then you can kind of prolong this time frame that you're able to recharge and pump out some more ATP, again, anaerobically without having to take in extra oxygen. And so there's a couple challenges with this. This will also help athletes gain some muscle mass. But what ends up happening is this actually ends up kind of giving your body a signal to kind of shut down its own synthesis. And the reason is that your body is really efficient, right? And so if all of a sudden you're taking in a bunch of creatine in your diet, your body's going to go, whoa, well, we have plenty of creatine. Why am I wasting my time, producing creatine? I'm going to go use that energy to produce other things. And so it can actually shut down our body's own synthesis of creatine. And this can, for some individuals, it just depends, again, on how sensitive they are and, their body's reaction to taking in creatine supplement. Also, how much creatine supplement that individual's taking in. If they're taking in a lot of creatine for a prolonged period of time, then this can make you more susceptible to, again, completely shutting down your body's own synthesis of creatine. And you could end up, there's some safety concerns with this, because you could potentially end up being stuck having to take creatine supplements for the rest of your life. Now, the other kind of challenge with this, I don't have this on here, but the other kind of challenge with this is that that extra creatine in your system is actually filtered through the kidneys. So this can end up putting an extra stress on your kidneys as well, especially if you're taking in really high levels of creatine. And so, this can end up causing some damage to your kidneys if you're taking high levels of creatine for a prolonged period of time. So again, that's kind of your first, go-to. And so when when your muscles are screaming, let's say you're doing a 100-meter dash, then your body's going to first go to creatine to supply its ATP, okay? Its second route, okay, that is going to go to is anaerobic cellular respiration, okay? So remember this process where we were, again, just doing glycolysis, basically, okay? And pumping out just little bits of ATP, okay? But that is enough to get you by for another 30 to 40 seconds, okay? So this will provide ATP for about 30 to 40 seconds. So again, this might get you through a 200-meter race, okay? But for those of you that were lucky enough to run the 400 like me, okay, somewhere after that 200-meter point, you do something called hit the wall, okay? And that's because you're going to need to kind of slowly transition over to aerobic cellular respiration, okay? Because you're just not getting enough ATP from the creatine and then the anaerobic cellular respiration. And it literally kind of feels like you're hitting a wall as all of a sudden your body's like, whoa, I need lots of oxygen. I need more oxygen, okay? And that's because you need to start to switch over to aerobic cellular respiration. And this production begins after about 30 seconds of like really intense exercise, okay? And once you hit about the 10-minute point of doing some pretty intense activity, you're going to rely on mostly aerobic cellular respiration. Again, it's going to provide about 90% of your ATP. So you're going to completely kind of almost switch over to aerobic cellular respiration for your ATP production. And if you're doing really prolonged periods of exercise, like you're running a marathon, again, you're relying on almost 100% then of aerobic cellular respiration to do those activities. So this is just kind of like a nice kind of demonstration of where you would switch over again to at these different points in terms of the different types of ATP production. So muscle fatigue. This is when you hit this point where you literally, your muscles have this inability to contract anymore because your activity has been too prolonged for your body in your body training, okay? So, and I say for your body and your body training because you can, again, as you guys know, train your body and train your muscles to endure kind of these longer, prolonged activities up to a point, right? But so some of the factors that are going to contribute to what we call muscle fatigue is one, first, this kind of what we call central fatigue. And this is really kind of more coming again from your brain. And this is this kind of general feeling of tiredness and this real desire to kind of stop exercising. And this is a protective mechanism that you have. This is your brain telling you, hey, I think we're about to push it too far and your muscles are going to start to tear. So I'm going to start to tell you that you feel tired. Okay. So that maybe you'll stop before you actually do damage to your muscles. Okay. You're also then going to eventually start to hit a point where you have this insufficient release of acetylcholine. So you're just not able to release acetylcholine fast enough. Okay. And you're starting to kind of deplete your acetylcholine stores, and you're not able to make that acetylcholine fast enough. And then eventually, obviously, you're going to deplete creatine phosphate. So again, you just can't recharge that creatine phosphate anymore because there's not any ATP to recharge that creatine phosphate because, again, you're using it all. And then you're going to eventually have a decline of calcium as well within the sarcoplasm. Again, because you're, again, just issues with lack of ATP there. And then insufficient oxygen or glycogen, again, because you're using up those as well. And then the buildup of lactic acid and ATP, okay? Or I'm sorry, not ATP, ADP, adenosine diphosphate, are going to kind of start to make your muscles feel, again, fatigued. So again, those are all causes of what we call muscle fatigue. Because again, if you've got lots of ADP, then you don't have a lot of ATP, okay? So we're going to stop there. And then on the next lecture, we will talk about how our muscles are going to work together and then what are the different types of contractions that can occur, okay? All right, so hopefully you guys found that interesting, and please let me know if you have any questions. I love answering questions about muscle contraction, so please send them to me and post them on the discussion board so that we can share them with everyone.